Okay, in this little mini lecture video, we're going to take a look at how enzymes provide alternative reaction pathways. So how do they allow chemical reactions to undergo different chemical steps to get to the same product that they would have gotten to normally in the absence of the catalyst? So the first thing that they can do is they stabilize charged intermediates. So charged intermediates that normally wouldn't form, for example, in a test tube in the absence of an enzyme can become more likely to form in an enzyme catalyzed reaction. So for example, they can stabilize oxyanions. So for example, in our last lecture video, when we bumped the electrons up to the carbonyl oxygen and we formed this negatively charged species, enzymes can even stabilize carbocations, which we know are rare to see. And you can stabilize these with negatively charged amino acid side chains, for example, for the carbocation. For the oxyanion, you can utilize positively charged amino acid side chains, hydrogen bonds, even metals can participate in this charge stabilization. So that's the first thing that they can do. The second thing that they can do is proton transfer for acid or base catalysis. So you've seen acid and base catalysis in test tubes before, but we can also transfer protons back and forth between the enzyme and the substrate. There are two different cases of acid-base catalysis. In the first, we call it specific. In specific acid-base catalysis, we utilize a water molecule or water molecules in the catalysis as either the H plus donor or the acceptor. Okay, so the H plus that's being moved around, the proton that's being moved around, is coming from a water molecule. So if water is functioning to donate that proton or to accept it, we call it specific acid-base catalysis. We can also have general acid-base catalysis, and in this case we're using amino acid side chains as the proton donor or acceptor, and that of course is going to depend on whether or not that amino acid side chain is an acid or a base. So we know then that the primary participators in this type of chemistry then would be are glutamate and aspartate residues. Those have carboxylic acid side chains, so they can serve to either donate their proton or the carboxylate can accept it. We've got lysine and arginine, which have positively charged nitrogens, which again, depending upon the state of that particular side chain, can accept or donate the proton. Histidine, we've also seen, can serve as a proton donor or acceptor, again, depending upon its protonation state in the active site. Then we can also, under certain conditions, see cystine's thiol side chain gain or lose a proton. And we can even sometimes see proton abstraction from the hydroxyl group of a serine or a tyrosine. So in general acid-base catalysis, you want to watch for the participation of these amino acid side chains. The third mechanism that enzymes utilize is called covalent catalysis. And in this situation, the enzyme actually forms a covalent bond with the substrate. So for example, let's say we had molecule AB and we wanted to hydrolyze it, so we wanted to break the bond between A and B utilizing an enzyme. So that might look something like this, where we have an enzyme with, say, a lone pair of electrons, and let's put a negative charge on that lone pair just to make it even more nucleophilic. We could have a nucleophilic attack at atom A, which would subsequently break the bond between A and B. So B would be our leaving group. And at that point, we would have a covalent bond between atom A and the enzyme. Then we could have a water molecule now come in, hence the hydrolysis part, and that water molecule could then do a nucleophilic attack at atom A, and our enzyme would be regenerated, and we would end up with a and our enzyme put back to the way that it started. So in this case, we formed a covalent bond as part of the intermediate steps of this particular chemical reaction. So that's an example of covalent catalysis. And you see in this process, in this hydrolysis process, it was the enzyme that broke the bond between A and B, and the water molecule that actually broke the bond between A and the enzyme, as opposed to in a test tube where it would be the water molecule breaking the bond between A and B. So those are the main types of catalysis, or ways that enzymes can facilitate alternative reaction pathways. And we'll do a nice examination of chymotrypsin to see a lot of these different mechanisms come into play.